When we want to get cells to make protein for us, we typically turn to bacteria first if we can, because they're cheap, they're easy, and if they work, they tend to work great, and you get great overexpression, so you get tons and tons of protein made. But sometimes you get tons and tons of protein made, and it's not actually soluble, because you're kind of getting the bacteria to make more protein more fast than they have time to fold them properly and stuff. And then your protein gets aggregated, and it forms these inclusion bodies, which are basically this insoluble clumps. And sometimes people go and they try to isolate the protein out of those clumps and kind of like denature it with urea and stuff and then refold it. Doesn't always work properly. It can be a lot of work. So there are other things that you can try to do in order to optimize the expression in bacteria. But at the end of the day, sometimes you got to switch to a different expression line. Bacteria are really great for small proteins that come from eukaryotic cells, so proteins that like already were made by bacteria. But if you've got eukaryotic proteins that are more complex, you've got things that have glycosylation, so sugar chains, they've got phosphorylation, all these various things. The bacteria doesn't have the same machinery for adding those post-translational modifications, those PTMs. And it doesn't have the same chaperones, so the same kind of like protector proteins that are going to help with the protein folding. And so things are different in the bacterial cells, and so they might not make those other proteins properly. So in those cases, you might have to turn to things like yeast, insect cells, and mammalian cells. And those kind of like increase in the amount of cost, time, and effort. And so often people will try to optimize that bacterial expression first if they can, if their protein seems to be one that would be amenable to that. Things you can try, first make sure, is it the expression or is it the solubility problem? And so with the expression, you can do a pre and post-induction sample on SDS-PAGE. See if you see a band show up in the post-induction sample that wasn't there in the pre-induction sample. That tells you your protein is getting expressed, but it's just not folding properly. If your protein isn't getting expressed at all, well, now you might want to go and check the plasmid sequence and make sure there aren't any mutations in there. You're also often just going to try it again, maybe on a smaller scale, but sometimes things just happen and you waste so much time trying to figure out what went wrong and you never find out. So just try again, and if it doesn't work now, then you want to go back to square one and try to re-examine everything. What can you change? Well, the first things first is kind of just the simplest things. Just slow things down. So you want to slow down the production of the protein so that the protein has time to fold properly. You can do this by lowering the temperature. And so maybe you lower the temperature to 18 degrees Celsius and do it overnight, because it's going to go slower so you get less protein. So you want to give it some more time. Other things that you could do is you could decrease the amount of induction. So say you're doing IPTG based induction, lower the amount of IPTG that you use for the induction. And those things are kind of things that you can do without having to change the actual construct itself. You can also change out the cell line you're using. And so what you can do is you can put it in a different cell line, a different strain of the bacteria that might have sorts of bells and whistles that are going to be better for your protein expression. This is especially important if your protein maybe has rare codons, and so it uses codons, spelling for amino acids that the bacteria doesn't use that much, and so it doesn't have enough of the tRNAs. And so you can use strains like Rosetta that have the extra codons, the tRNAs for rare codons, extra copies of them, and so then those cells will have more of those tRNAs that you need to make your protein. There's also strains if your protein is kind of toxic to the cells. There's also ways that you can kind of get your protein secreted instead of having it be intracellular, or you can make it in various compartments inside cells if your cell's toxic. And so there's some cool tricks. Also, if you're dealing with toxic protein, you want to make sure that you keep the expression level low and that you don't express it until you're really ready. And so you don't want there to be a leaky promoter. And so there are some things like some bacterial strains that have additional things that are going to make it so that you don't get that early expression, as well as you can use different promoters and stuff, but that requires switching out your vector and not just changing the growth conditions. So those are all things that involve changing just the growth conditions, but there are also things that involve changing the vector. So changing like the plasmid or even the protein within the plasmid. So like the construct that's encoding the protein. First though, let's deal with what you can do with the stuff that's before the protein. Because we were talking about slowing things down, decreasing expression. You can use a different promoter that is going to then be not as strong of a promoter, and then you'll get lower level of expression, and that is going to allow you then to have your protein, give your protein more time to fold. If your protein is toxic, then you're going to really want a tight promoter, and so you want to keep the leakiness away, and so there are promoters that are really good for not making your protein until you're really, really ready. And then there are things that we could do to actually change the protein itself, or hook up stuff that's attached to our protein to try to get our protein to be made. And one thing that you could do is you could add a fusion partner. So a fusion partner would be something like Sumo, GST, MBP. These are small soluble proteins that you put in front of your protein. And now the bacteria, they really like making these fusion proteins. And so they'll be happy to make the fusion proteins. And then your protein's attached to it, so you get tricked into making your protein. And because these little proteins are really soluble and nice, they're going to help your protein be soluble and nice. And they're going to kind of protect the non-polar parts of your protein as it's getting made, and kind of act as chaperones to help with your protein be folded. You're often going to have a protease cleavage site in between the fusion partner and your protein that you can cut off later, and then hopefully your protein is still soluble after you cut it off. You can also, going back to the codon things, you can do codon optimization of the gene. So say you're taking a gene or the coding sequence, so the CDS, so the protein coding sequence for a gene, and maybe it's from humans. And so it's going to use the codons that are common in humans. You can use codon optimization to change the spellings to those that are common in the bacteria, but then you've got to kind of like redesign the whole gene. You can also try, if you're using a tag for affinity purification, you can try like moving it from the N terminus to the C terminus. You can try changing around the linker length. All these various things which seem like they might not make that much of a difference, they actually can make a difference in the expression. You can also take off parts of the protein, so you can like truncate regions that are expected to be really floppy and loose and intrinsically disordered and stuff. Just note that when you're making these changes, you might actually be changing the functioning of the protein. And so whenever possible, you want to have some sort of functional test to make sure that any changes that you're making to the protein aren't actually going to mess up what the protein is doing. And then if all else fails, then you might want to think about changing to one of those other expression systems. Or if you know from the beginning that it's probably not going to express well in bacteria, then you can just start in those other expression systems. There's also strains of bacteria that express additional chaperones, so proteins that'll help your protein be folded. But again, these bacteria are still lacking a lot of the machinery for doing things like post-translational modification. And so for those, then you're going to want to go to those more complex cell types, which again is a lot more work. You need a lot more equipment, you need a lot more money, and you get a lot less protein. But you do what you got to do. And so that's why you might see that there's a lot of pharmaceutical things that are made in like CHO cells, so Chinese hamster ovary cells. There are things made in 293 cells, which are a type of human cells. Then I used insect cell expression, so with baculovirus, BEVs in grad school. Those are great. They're expensive and time consuming, but they're cheaper than the mammalian cell expression systems, but they do have slightly different glycosylations. There are also different glycosylations that you'll get with yeast cells. Yeast cells are more similar to the bacterial cells in kind of the scale that you can do things and the ease, but they're very hardy, and so they're like harder to break open the bacteria and stuff. And so they're not as easy as bacteria. They're also cell-free expression or CFE systems, and this could be based on lysates or it could be based on purified components, so like the pure system. And this can be good if your protein is toxic, but we're typically dealing with lower levels of your protein that get made then, and high expense. With any of these expression systems, you're typically dealing with cells in a flask, so a suspension culture, as opposed to cells that are stuck to a plate, so adherent cells. And this is going to allow you to make a larger amount of protein than you could if the cells were stuck to a plate. But sometimes the protein's expression systems, if you want to make it in a very particular cells, people might still make it in those adherent cells. And often people will start with adherent cells when they're doing the scale up, because it's easier to change out the media for adherent cells and then having to spin things down and remove the media, but then they'll kind of ramp up to larger sizes and switch over to those suspension cultures. So bacteria are typically like your first go-to choice, and then if you got to go somewhere else, then you, depending in part on what you have available to you, then you'll go to one of those other systems. But you can try your best to optimize that bacteria, change your temperature, change the IPTG, change the growth time, change the cell line, change the plasmid, change the spellings, all these various things that you can do to try to get your protein made from those bacterial cells if you don't have all that other stuff, or if you just want to keep things cheap and easy. So good luck.